Good morning, everyone, and welcome to this webinar. My name is Sarah, and today I will be presenting you our smarter solutions for RNA-seq from single cells and other challenging samples. Just before I start, if you have any question during the webinar, please type it in the chatter box and I will take them at the end of the presentation. Okay, so in today's presentation I will start talking about the challenges of single-cell RNA sequencing, and then I will explain how our SmartSeq technology has helped to overcome these challenges. Then I will talk about how we have adapted this technology to enable RNA-seq from other challenging samples, and I will focus in 4 specific applications: degraded RNAs, small RNAs, T-cell receptor profiling, and targeted RNA sequencing. And I will finish by briefly mention how we have expanded this smart technology also to the sequencing of DNA for some specific applications like ChIP-seq and MET-seq. So let's start talking about single cells and why it's important to do studies in single cell. As you may know, the gene expression can vary significantly between cells, and when you are measuring a population, you are measuring an average of the expression, which not necessarily will correlate with the expression at the level of each single cell. So this is illustrated by this paper in which a single cell sequencing was performed in cells from bone marrow. And you can see here in blue the splicing patterns and expression levels from some specific genes in different single cells. And in gray, you have the expression for the bulk of 10,000 cells. So you can clearly see that for some genes here on the left, the pattern of expression of these genes are very similar between the single cells and the bulk population. But for other genes, there are clearly differences, like highlighted here, but you can see it also in other places. There are clear differences between the pattern of single cells as compared to the bulk population. And actually what has been demonstrated now by several publications is that these cell-to-cell differences can be critical in many physiological processes, but also in diseases. So doing single-cell RNA-seq is very challenging, and of course because of the very small amount of RNA which is available in one single cell. You can estimate that the amount of total RNA in one single cell will be of around 10 picogram, but of course this will depend on the cell and the physiological state. And also most of this total RNA will be ribosomal RNA, so the fraction of coding RNA or messenger RNA will be even lower. So a performance single cell method will need to be first sensitive to enable to do the cDNA synthesis from these trace amounts of RNA. Then it should be reproducible so that the technical variation will not hide the biological variation, which is what is of interest to you as a research. Then, of course, it's important that the method enables to take cells as direct input to avoid RNA purification, which can be impossible to do from one single cell, and also require minimal handling to limit the sample loss, degradation, and contamination. And finally, it should be able to yield full-length cDNA in order to maintain the transcript length and the representation of the transcript in the original pool. So all these features we have incorporated in our technology for ultra-low input RNA called SMARTer. And let me explain you a bit more in detail how it works. So we have here the general workflow of the latest generation of smart technology, which is the SmartSeq V4 ultra-low kit, and how it works. So you will start a reverse transcription with an oligo-DT primer in this case to only amplify mRNA. The reverse transcriptase that we use has a specific property, and it is that when it reaches the 5' end of the transcript, it's able to add some non-template nucleotides that you can see here represent like X. And this reaction is done in the presence of oligonucleotide called the template switching oligo, because this oligonucleotide contains bases that will base pair with this non-templated nucleotides added by the RT. And then it will allow the RT to switch the template. It means to switch from using the mRNA template to use the sequence in the oligo to continue the reverse transcription. So what this enables is to obtain a double first strand cDNA with known sequences at each of the ends, and these known sequences will be used to amplify the cDNA by PCR, and then we will get a double-stranded full-length cDNA. So the advantage of this method is that it can work directly with cells. So you can start with as low as a single cell and up to 1,000 cells. But also you can start with total RNA with as low as 10 picogram. As I said, the method used here is oligo-DT priming. So there's no need for ribosomal RNA removal or for DNase treatment required. And this is important because you can imagine that in a cell lysate you will have both ribosomal RNA and DNA. The method is a single tube workflow, so limiting risk of contamination and degradation. And it will generate full-length cDNA, which is compatible for library prep with Ion Torrent or Illumina platforms. And you can see here electropherogram, the typical trace that you can observe when using the SmartSeq V4 method with human sample. You can see that we obtain full-length cDNA between 400 base pairs and up to 14,000 base pairs, and with a peak at 2 kb. So this full-length cDNA needs to be processed before being able to sequence, because for the sequence we need to cut this in small fragments and add adapters depending on the platform. So we summarize here the three alternatives that we offer depending on the platform. So if you are using Ion Torrent, you can do enzymatic sharing and then add the adapters using the Ion Express library prep. If you are using Illumina sequencing, you have two options. One is use covariance sharing, so to mechanically fragment this full-length cDNA, and then use our low input library prep kit to add the Illumina adapters. Or you can use the Nextera XT protocol, which does something called fragmentation, meaning that it will simultaneously fragment the cDNA and add the Illumina adapters. Also, we have adapted this technique for allowing to use it in this instrument, the Fluidigm C1 system. So the C1 system is developed by a company called Fluidigm, and what this instrument does is that it allows to automatically capture single cells and distribute them in a sort of microfluidic chip, and it will also in an automated way inject all the components necessary for the lyase, reverse transcription, and PCR steps. So we have this specific kit that will provide the reagents in the appropriate volume for the use with this instrument. So you have noticed that in the name of the product we are talking about version 4, and this is because the first version of this single cell technology was launched around 5 years ago, and since then we have been doing continuous improvement on the technology to make it as sensitive and as performant as possible. And for example, in this fourth version, we did a big improvement, which was incorporate LNA, which means locked nucleic acid, on the template switching oligo. And this, in addition to all our experience in improving the template switching reaction, has led to a very performant version in which we obtain reduced background, and also we have considerably improved sensitivity and reproducibility. And let me show some data about it. So you can see here a comparison only using 10 picograms of mouse brain RNA, comparing our previous version with current SmartSeq V4 version for 2 replicates. So the first thing that we see is that we can reduce the number of cycles, showing that it's more sensitive. And even doing that, we are able to identify around 30% more of genes with the before technology. And also we are able to do this keeping all the other metrics very good, as it was the case already in the previous version. We have an excellent mapping to the genome and to exons, and allow mapping to intergenic regions. So we saw in the previous table that the results between the 2 replicates were very similar, but another way to look at reproducibility is to analyze these scatter plots, in which the expression levels of each transcript in the 2 different replicates are represented here in this graph. So we can calculate the coefficients of correlation between the 2 replicates, and the closer they are to 1, the best is the reproducibility of the assay. So you can see that with the SmartSeq V3, so the previous version, we have a good coefficient of correlation when we analyze the whole population, and it's the same for SmartSeq V4, but we have a better sperm correlation, so we have increased the reproducibility overall. But this is more obvious when we analyze the low expressed transcripts. So shown here in this zoom on the left, you can see that the dispersion is much higher with the previous version, and this is reflected in this low coefficients of correlation, while with SmartSeq V4 we are getting much more reproducibility. We also measure the body coverage, the gene body coverage on the transcripts, and for this experiment we use single cells. As you can see here, we took the 6 single cells in 2 different body populations of 100 and 1000 cells, and you can see that very nicely the gene body coverage is very uniform along the transcript. But sometimes if you are interested in analyzing only differential expression between 2 populations, you don't need to have the full length information, because for this kind of analysis only getting a part of the transcript, for example the 3' end, will be sufficient to give you the differential expression information that you are looking for. So we have developed a new application for the SmartSeq V4, and this is this kit, SmartSeq V4 3' DE kit. So DE stands for differential expression. And 3' is because in this kit we are only capturing the 3' end of the mRNA, as you can see here. So differently from what we observed in the previous slide, we'll have the uniform body coverage up to the 5'. Here we are only keeping the 3' end. This kit is a single cell capable, and actually this data shown here is from different cells as compared to a pool, as you can see here. And or you can use it also with up to 100 cells or up to 1 nanogram of RNA. In this kit we have enabled pooling samples, and you will be able to use 12 different barcodes in the oligo-DT to enable this pooling before going to the library prep. And then by combining with the different Illumina high throughput indexes, you will be able to multiplex up to 1152 samples per sequencing lane. And for this specific application, we provide a specific tool which is available on our website to do the multiplexing and analysis of differential expression. So this technology, smarter for single cell, as I say, has been there now for some years, and since then it has been used in many different cell types and many different applications and with different methods. You can get your cells by manual isolation, by fax, also by laser capture, or using the Fluidigm instrument that I mentioned before. But also it's interesting to know that it's not only for starting with cells, because we have validated this protocol for single cell using mammalian cells, but you can also use it starting from RNA, and if you are working with plants, with yeast, with other species, it's also feasible to use this technology. It's just that in this case it's better to extract the RNA. And there are many publications supporting this. But not everything will work with this SmartSeq V4 technology. There are some samples that we know for sure that won't work, and these are low quality RNA samples, means degraded RNA, or non-polyadenylated RNA samples, because the method is based on oligo-DT priming. So this leads me to the second part of the presentation, in which we will discuss how we have enabled to work with these challenging samples. So in the first case, if you have low input RNA and it is degraded, usually it won't have a poly-A tail, because the RNA degradation will start from the ends. Or if your RNA is naturally non-polyadenylated because it's from bacteria or from a virus, you can't use oligo-DT priming. So in that case, what you can use is random priming, and here is the method that we have adapted to use the SMART technology with random priming. It's called Smarter Stranded RNA Seq Kit. And as you can see here, you can recognize the general workflow that I showed before, but we will start using a random primer or N6 primer. And also, since this RNA will be already cut into pieces, because we are starting from fragmented RNA for this protocol, we can incorporate during the PCR steps the Illumina indexes and adapters. So the advantage of this is that you can start from your RNA sample, and finish with a library ready to sequence in Illumina platform in few hours. This is very fast, less than 4 hours. Also, another advantage of the method is that it provides inherent strand information because the smart reaction is directional inherently, so and without using the DUTP method. The input for this kit is 100 picogram of RNA, but it needs to be depleted from ribosomal RNA, because you know that when you are using random primers, you will also reverse transcribe ribosomal RNA, and this you don't want to have in your sample. So if you have mammalian samples, you can use ribogone mammalian that we have developed specific for accompany this smart stranded kit. The minimum input for this ribogone mammalian is 10 nanogram of total RNA. But what happens if you have less than 10 nanogram of RNA, and so you can't use ribogone mammalian to do the ribosomal RNA depletion, and you can't use another method because there is no method currently to do this with less than 10 nanogram. So in this case, when you have these pico amounts of RNA, we have developed a new solution called the Smarter Stranded Total RNA Seq Kit Pico Input Mammalian, which is compatible with as low as 250 picogram of mammalian total RNA. And the originality of this kit is that instead of trying to deplete the total RNA from ribosomal RNA, we are going to do a reverse transcription of the whole population in this PICO kit, and then doing the depletion of ribosomal sequences on the cDNA level. So this is what is shown here. We will use for that proprietary technology called SAP-R and R probes that will specifically target the cDNA derived from ribosomal RNA. And as a result, we will have a library that will be depleted on these ribosomal cDNA sequences. So this method keeps all the advantages of the the regular stranded technology. So the workflow is very fast. You go from total RNA to library ready to sequence in only 5 hours. You keep strand information. And the advantage of this kit is that you can use RNA of any quality with RIN as low as 2, and use very small amount of RNA. So here we are showing some data obtained with this kit, comparing different input amounts, so from 10 nanogram and up to 250 picogram, which is the lowest recommended amount. You can see here that we have also tested a lower amount, in this case 100 picogram, and interestingly, the number of transcripts that we identify, or the general metrics, are very similar regardless on the input amount. But it's just that when you use very small amounts, you need to be aware that some metrics will not be as good. Like for example here, for 100 picograms, you can have a high duplicate rate. Another important thing that you can observe here is that percentage of ribosomal RNA left is relatively high, so around 10%, but I say that it's relatively high as compared as what you obtain using a ribogone, because with ribogone you will get always less than 1% depletion. So here, yes, you will have more ribosomal RNA left, but this is much better than what you will obtain if you didn't do this ribosomal cDNA depletion, because you will have 90% of your ribosomal cDNA. So here we did an experiment using a very hard sample. In this case it was a FFP sample from human liver, and in this sample we used the method with the 3 different input amounts, and you can see that we are always keeping at least 35% of ribosomal RNA. So this is a particular sample which is very hard to deplete. But when we compare to the results when we don't use the ZAP-R and the R probes, we can see that we have 90% of reads that align to ribosomal RNA, and more importantly, we are detecting 3 times less transcripts that when we use the method. So it shows that this method will really allow you to increase the sensitivity and to identify more transcripts. But this PICO kit allows the efficient capture of fragments of a minimum size of around 100 nucleotides, as we shown here with 2 different RNAs. But what happens if you are interested in analyzing RNAs smaller than that, smaller than 100 nucleotides? So you are interested in analyzing small RNAs. We have developed also smarter technology to enable working with these RNAs. So this is called the Smarter Small RNA Seq kit for Illumina. And it will allow you to create a library from small RNAs in a size range of 15 to 150 base pairs. So this includes, of course, many physiological microRNAs, but also it can be used for other applications that typically generate RNA which is very fragmented. For example, RNA immunoprecipitation or CLIP or ribosomal profiling. The input of this kit is only 1 nanogram, and what is nice about it is that you can start with total RNA or with an enriched fraction if you wish, but if you only have 1 nanogram of total RNA, it works perfectly fine. The protocol is also gel-free, and by gel-free it means that you don't need to do this page purification that most of small RNA protocols require. We provide 2 options for the size selection. One is using impure beads, and the other one is using a blue pippin machine. And the method is ligation free, and this has an advantage in the detection of the small RNAs, and this I will show here in this slide, because actually the traditional ligation methods that have been used to analyze small RNAs, it has been demonstrated that they have some bias issues, and this is because the adapters that are using to ligate the microRNAs have differences in the efficiency of ligation depending on the microRNA sequence. So in this experiment we use a synthetic library of almost 1000 microRNAs, and they are all at the same concentration in this synthetic library. So we prepare a library using the classical adapter ligation method or our smarter method, and then we measure the level of expression. So since all the microRNAs are present in equal quantities, we would expect that the normalized expression level would be around 1 for all. We should have normally a flat curve here. But this is not that what we see. We see for the adapter ligation method that actually only 22% of the microRNAs were found within a 2-fold cutoff on this expected expression level. So this is reflecting these differences in the efficiency of adapter ligation. With our smarter method, we are able to obtain 55% of the microRNAs that fell within this cutoff range. So it shows that our method is at least 2.5 times better than the adapted ligation method, and that the expression of microRNAs will be more representative using smarter methods than with adapted ligation. Okay, so another challenge in samples is the T-cell receptor repertoire profiling. Very briefly, the T-cell receptor are heterodimeric receptors, a protein composed of two subunits that are at the surface of the T cells, and they are highly specialized for antigen recognition. So the TCR alpha and TCR beta chains are encoded at two different loci, and here I am only showing the TCR beta DNA locus. In what is very well described and known now is that this TCR locus undergoes a very important recombination during the germline and then during the splicing. And the idea of this is to generate very high diversity of TCR receptor that will be able to recognize any antigen. So there are hundreds and thousands of different TCR sequences, and this is what is called the TCR repertoire. And the challenge here is to be able to sequence all this huge diversity. So what people are currently using for doing that is mainly multiplex PCR approach, using primers that are designed against the different regions of the TCR locus. But this, of course, is susceptible to amplification biases, and you can't get the full length region of this variable region. And also you can analyze only the alpha or the beta chain on a single experiment. Another way of doing that is using a race approach, in which you will start from an mRNA-derived template, so you will be closer to the actual protein sequence when you start from mRNA than when you start from gDNA. And the race actually will allow you to get the full sequencing data for the whole variable region. You only need to design primers against the constant region, and you will be able to get the 5' end of the transcript without knowing the sequence. And you can also put primers for the alpha and the beta regions in the same experiment. So this is a method that we have adapted in our smarter TCR alpha and beta profiling kits. As you can see here, you can recognize the general smarter protocol. And we have designed specific primers against the TCR alpha and beta sequences, and this allows us to get the full TCR alpha and beta sequences for human and mouse. These are two different kits because the sequences are very specific. Both kits are able to work from either purified RNA or also from purified T cells. And here I'm just showing some data for the smarter TCR kit, in which you can see that analysis that were done using splenocytes or CD4 plus T cells, and RNA was extracted from these samples, and we can see here the mapping to the TCR alpha and beta sequences, that we get a good on-target mapping and with a similar proportion between both of the subunits as it could be expected. And also here we can see a saturation curve of the number of reads that are required to, as compared to the number of clonotypes that can be identified. You can see that the number of clonotypes identified can vary between 10,000 to 40,000, and this of course will depend on the sample. But it's interesting to see that the assay is reproducible between the different cell populations. You can see splenocytes that have a similar level of diversity all four together as compared to these CD4 T cells. So the last challenging sample that I will be mentioned today is rare transcripts, and let's say that you are interested in studying a transcript that is very rarely found, or some specific alternative splicing isoform that has very low frequency, or a rare event like a gene fusion. So when you have this kind of rare transcripts that are difficult to identify in a whole transcriptome analysis, you can decide to do a target sequencing approach. So this means you can only do a library on the transcripts of interest. And this is what we propose here with the smarter target RNA capture. This kit will allow you to generate a library only on the genes of your interest, and it will be you, the researcher, that will decide which are the genes I am interested in, and you will design the probes to capture these genes, and you will be able with this kit to pull down the RNA of interest and not the cDNA as is done in other enrichment methods. So you will capture your RNAs of interest, and then using SmartSeq v4 technology, you will be able to get the full-length transcript information and prepare a library only from these transcripts of interest. Since we are targeting RNA and not cDNA, this has many advantages. So the workflow is shorter. The hybridation is very fast. It's like less than 1 to 3 hours. And also the probe design is very easy. You will only need to design one probe per exon to do this. Now here is just an example of the data that you can obtain. In this case, we spiked in a fusion for this gene, BCR ABL1 fusion, and we spiked this fusion in this human brain RNA at different concentrations, and we wanted to see at what concentration we were able to detect the gene fusion. So the probes that we designed were only against the ABL1 gene, so only one of the partners of the fusion. We didn't do that for BCR, the other partner. And what we see is that if we don't do any capture method, we are not enriching the transcript, and we find very few reads that support the fusion. But when we do an enrichment using our target RNA capture method, we're able to enrich at least 150 times this ABL1 gene, and we can see that at 1% of spiking we are able to detect reads supporting the fusion. So of course you can imagine that if you use also probes against the older partner of the fusion, you can enrich this much more. So this shows us that our method enables to detect a gene fusion that can be present at a frequency lower than 0.5% when we are targeting only one of the partners. Okay, so we are arriving to the end of the presentation. I will just briefly mention that we have adapted also the SMART technology to DNA because we have seen that smart technology is very powerful when starting with very small amounts of RNA, and we saw that we could also use this with DNA applications that generate very small amounts of DNA. So this is the product that we have, is DNA Smart Chip Seq kit. So it's very sensitive. You can start with only 100 picogram of DNA, because if you have done chip, you probably know that it's very common to obtain very small amounts of DNA. And also, most of the time, the DNA can be single-stranded, and this is nice about this protocol, is that it can take both single-stranded DNA and double-stranded DNA. So the protocol is very fast, because actually this is an adaptation of the smart technology to be able to take a DNA. We are doing just a poly tailing to the DNA in order to allow the annealing of DNA Smart Poly DA primer to do reverse transcription from the DNA. Well, reverse transcription is a strange term when talking about DNA, but is reverse transcriptase that will be copying this DNA and then doing the smart reaction that I already mentioned. And we will incorporate the Illumina indexes and adapters by PCR, and then you will get your library. So very recently we have also applied this technology to enable the analysis of genome methylation, and more specifically to do MBD-seq. So MBD stands for methylated methyl binding domain protein, and these are proteins that interact specifically with methylated DNA. So in this case, in the specific case of the EPIXPLORE METH-seq DNA enrichment kit, we are using MBD2 protein, which is known because it interacts with a high affinity with CpG island in the DNA, in the methylated DNA. And it's actually a very rapid method in which you will start from shared genomic DNA, and you will incubate this DNA with the MBD2 protein, which is His tag, and then using another of our technologies called capturing technology for separating protein fractions, you will be able to separate the methylated fraction from the unmethylated fraction. And then once that you have your two fractions, you will be able just to use the smart technology to generate the library. So this is shown more in detail here. Just in 2 hours you are doing the part of separating the fractions, and then you have exactly the same DNA Smart protocol that I mentioned before to generate the library. Okay, so here I had to present not in much detail the data, but we have a lot of data available for each kit. You can go to our NGS resource portal, and for each kit we have tech notes, we have webinars from the R&D developers of each kit, we have scientific posters, FAQs, and citation lists also if you want to see more examples. So do not hesitate to consult this resource portal. And now I will be happy to take any questions that you may have. So if you have any question, please put on the chat box. Okay, let me check. I have a question. May the smarter kits be used to create a library from RNA collected by patch clamp. So, well, I'm not very familiar with this technique of patch clamp. I would say that as long as you are able to get RNA from your experiment, or cells that are not fixed, because that's important to keep in mind when you are starting directly from cells. It's important that cells have not been fixed, because this is not compatible with smarter RNA synthesis. So cDNA synthesis, sorry. So if you use some kind of fixative for do this, you can't use cells directly, but what you can do is extract the RNA, and if it's of good quality, you can use with a SmartSeq V4. If it's of degraded quality, you can consider using a Pico kit or the stranded kit, depending on the amount. So I have another question here. Can be the small RNA-seq kit used with plasma samples? So yes, we have a poster on the website in which we show data done in collaboration with researchers from the University of Massachusetts. And what we showed there is that the method could be used with RNA isolated from plasma and serum. Small RNA libraries prepared from this, and this is interesting for people that are interested in biomarker discovery, and yes, working from these samples. We give also in the poster recommendations on the method to use to extract the small RNAs, because we have seen that depending on the method used, there can be some differences in the efficiency. So please refer to this poster for more information. Okay, another question here, if SmartSeq V4 can be used with Drosophila. Well, as I said, if it depends for using the cells directly as input, you can, we have only validated this with mammalian cells, because actually we are using a lysis buffer, and this lysis buffer might not be strong enough to lyse other cells that can be harder to lyse. So it depends of if your cells are easily or not easily lysed. But what we know is that it has been used from using RNA purified from the cells, Drosophila cells. As long as the RNA is polyadenylated, so any eukaryotic RNA can be used with the SmartSeq V4. As I say, if the RNA is not polyadenylated, like in the case of bacteria or virus, then we recommend the smarter stranded kits that are based on random priming. Okay, any other question? Is a TCR compatible, TCR profiling kit compatible with other species? So no, the kit is very specific. The mouse kit cannot be used with the rat kit, because the sequence of the primers are very species specific, so it's not possible just to take another primer and use the TCR. So if you are working with human, you will need the TCR human kit. If you are working with mouse, you will need the mouse TCR kit, and so far we don't have for another species. Okay, so if there are not any more questions, we are closing the session. Thank you very much for participating, and if you have further questions, do not hesitate to write us to techeu@takarabio.com. You will get the address. Thank you very much. Bye.